Thank you very much for the kind introduction, and thank you for the invitation. Thank you, Matthias, for allowing me to be here. Matthias and I share several levels of connection. One, of course, is we both do biomedical research in Munich on the north side, on the south side. We share the first name, but we also share love and passion for metabolism. And I do have to make one disclosure. I know actually, in spite of what was just said, nothing about proteomics. If I have to do anything in that direction, I call Matthias or other collaborators. And we have recently collaborated, and very interesting things are developing, but that's still very, very early. When I was invited, I asked, well, really, you want me to be there? Because I talk about metabolism and diabetes and drugs, and that's very different. But it was confirmed that I should do that, so I'll bother you the next 20, 25 minutes with what we have been doing in the recent years, and maybe there's even a connection to, I think it's maybe this one, to proteins at least, if not proteomics. All right, that looks good. So of course I had to start with a beautiful picture of Matthias, looking good right there. But really this is about the molecules and novel drug candidates we have been trying to develop to stop a terrible disease that's called diabetes. And I'll take you back initially 100 years, almost 100 years. In 2 years there's the centennial anniversary of the discovery of insulin. That has certainly changed the world, right? Insulin, it turned a deadly disease into a manageable chronic disease, transformative. Now, it was a different time. Maybe these types of major discoveries were more easy to make, and by the way, what was different too, you could make a discovery in 21 and get the Nobel Prize in 22. That sort of has changed too. Now, if you jump until today or yesterday, a few years ago, the official associations in Europe and the US and worldwide on diabetes recommend how we deal with diabetes still the same cornerstones. If you look at this, well, give insulin, give metformin that we have known for 55 years, and maybe you can go back to Hippocrates even, diet, exercise, and education. This is what we do. So in spite of this transformative event 100 years ago, we really didn't continue as a research community to deliver other solutions. Well, maybe this is because diabetes isn't really a problem anymore. That's not the case either, right? If you look at epidemiology, we've heard already about epidemiology today, it's more than ever, almost 10% diabetes today in this society. It's going to go further up, hundreds of billions of annual global cost, 6th leading cause of death. Diabetes kills more people every year than AIDS, tuberculosis, malaria together across the world, globally. Now, that needs to be stopped because our society won't be able to sustain our lifestyle, and this is how diabetes looks. It's not just a lifestyle disease, it's the leading cause for dialysis, blindness, and so on, and it concerns children as well as adults. So I hope I have woken you up this afternoon with these horrible pictures and got you interested in how we're going to prevent this terrible tsunami or this catastrophe that 10, 15 years from now, we will have lost. We need solutions now. So one of the drivers of this is this sort of harmless looking set of curves, obesity. Nobody really understands, after going to these conferences for 30 years, nobody really understands how obesity may drive type 2 diabetes, and type 2 diabetes is like 95% of diabetes. But certainly everybody's convinced if we wouldn't have a global obesity epidemic, we wouldn't have that level of a global diabetes epidemic. Right? So we could stop that. Why don't we do that? Well, there are still some that say, not really. Obesity isn't a disease either. That's a lifestyle thing. Don't worry about it. If you look at obesity trends in life expectancy, this is the United States, and it's not Republicans and Democrats that's obesity in life expectancy, but it totally overlaps. And there are smart people who use smart mathematics that I'm not capable of that say, well, this means years of life. You may lose 7, 10, 14 years of life because of obesity as a global society. Well, that does matter. That makes you think. Do we have drugs? Yeah, we do have drugs. Some are approved and there's some more on the horizon. But if you really look at them and you consider the placebo effect, you may lose 5, 7, 8, 9% of body weight depending on which subpopulation you're part of. It's not going to make a dent in the global epidemic. We need better ways medically to deal with this. Now, there was a lot of excitement when I got into research. I was a clinical endocrinologist here in Munich in the University Hospital when this paper came out. Jeff Friedman, who's now a close friend and member of our scientific advisory board, discovered that there is a hormone called leptin that regulates body weight. That was unknown until then. And he discovered, well, adipose tissue is actually an endocrine organ. That was new, and that's basically why I got into research and thought, well, there's something I can do that goes beyond just giving insulin. Well, it turns out, even though leptin is a hormone that you can inject in mice or give humans that have leptin deficiency and then correct obesity, that wasn't the solution either, because most obese individuals, 98%, have high leptin levels. They have leptin resistance, sort of like insulin resistance in type 2 diabetes. That didn't solve the problem. But it taught us a lot of important things. One of them being that the brain is part of this. We need to understand how these hormones act on the brain, because 95% of what leptin does, it does by acting in the brain. Brain regulates body weight and glucose metabolism in response to leptin. When I was a postdoc and worked at Eli Lilly, of all places, 25 years ago, we then ran into the observation there's another hormone coming from the gut, from the stomach, called ghrelin. That's the opposite. Increases hunger, increases appetite, increases body weight, works at the same places in the brain that leptin does. But again, just blocking ghrelin didn't do the job. Not enough. Not powerful enough. Still taught us the same lesson. Somehow the brain is part of what obesity is all about and what type 2 diabetes, therefore, may all be about. If you then take a step back and look at the entirety of the genome-wide association studies, the GWAS studies, and I used to be skeptical and maybe to some extent still am, because that's a lot of money that went into them. But if you look at not the single results, like which gene is now responsible really for that disease, but the big picture, you find that the majority of hits related, for example, to obesity are related to proteins that are in the brain. And there is newer studies with even more individuals examined. It looks like obesity is a brain disease based on the entirety of the genomic information. All right, so overall, that led for us, when we then started to say, we want to go a step further. We don't want to just do descriptive work and inject insulin. We want to find a way to stop the obesity epidemic. We said, guiding hypothesis number one, the brain somehow needs to be a target. All right? How to do that? Well, has been tried before. Actually, some of you may remember, many years ago, some years ago, there was a drug that was approved in Europe and was on the way to be approved in the United States that blocked the cannabinoid receptor type 1. Did good things to obesity and appetite and metabolism, but had side effects, cardiovascular effects, but also caused depression and suicidal tendencies. Well, it turns out that those of you who may have used agonists for the cannabinoid receptor type 1 don't do that for appetite, but for other purposes. So it was not that surprising that there was depression as a side effect. But it revealed an issue that we then had to tackle. We want to target the brain, but based on today's knowledge and technical approaches available, you can target a specific neuroendocrinological pathway or neurochemistry pathway just in the part of brain you'd like to target it in order to set populations of cells. That never works. We don't have the specificity. We can't say, I don't want to go to amygdala and cortex and other parts of the brain, but I want to go to the hypothalamus. Specifically, this nucleus in the hypothalamus. And I want to change dopamine or serotonin or AGRP or NPY there. Therefore, our approach was different. We said, okay, can't do that, too dangerous, going to come with side effects. We're going to take an indirect approach. We use Mother Nature's toolkit. Peripheral hormones that are designed to respond to nutrients, to environment, to changes in metabolism that know where to go in the brain to then affect metabolism. So that was the second part, but then which signals should that be? We had a hunch it should be several, because everything we tried as a single signal didn't work, wasn't powerful enough. It was clear over time that there was a pattern we needed. Well, there's so many. Even if you just look at gastrointestinal signals and the most important ones, this is how it looks. Which ones are you going to go with? And knowing what it takes to develop drugs, if you want to go through some stages of that, you can combine all of those in all kinds of patterns. That may work in silico, it won't work in the lab. So we took a lesson from an unlikely place. Surgeons, I hope there's no surgeons in the room, but surgeons taught us that, well, they know how to treat obesity. Morbid obesity can be treated pretty well with bariatric surgery, a gastric bypass that you see being done here, or sleeve gastrectomy, something similar. Now, what not all of you may know is that right after the surgery, many patients can throw away their insulin. Their metabolic improvement is tremendous, and they haven't even lost any weight yet because gut-brain signaling is programmed differently after the surgery. It doesn't work for everybody, but for the majority, and it works not for everybody forever, but it taught us important lessons about something happens there that disconnects nutrient sensing with brain awareness of the metabolic situation that sort of could maybe be mimicked, we thought. What if we find the right signals that are involved here and reinvent them as a medical therapy, an injectable, something that you can swallow, and the brain thinks there's bariatric surgery, but there really isn't. So that then completed our main set of hypotheses about 15 years ago, and where we said, well, okay, we need polytherapy, and we need to take a lesson from which signals are involved in bariatric surgery. And if as a ex-clinician turned physiologist, pharmacologist, you want to make drugs, what you need is a chemist. And for the younger colleagues among you, I would really like to encourage you to take that step at whatever point in your career and commit to interdisciplinary research. It does tremendous things, but it is painful, and it requires years of learning each other's language. But I had the pleasure of meeting Richard DeMarchi in the 90s of last century. He a peptide chemist, and we then reconnected later after both of us went from industry to academia and said, well, we can do something together. Let's try to make drugs against obesity based on these hypotheses. So we started somewhere, and there's a number of hormones that we knew came from the gut, talked to the brain, changed with bariatric surgery. And you have heard Nikolai talk today about GLP. There was a starting point. We said, well, there's drugs out there that do good things, just not enough. It could be a starting point. Why don't we start with GLP, but look at other members of that hormone family? If you do that, you realize when you're a peptide chemist, there are similarities among the hormones that are part of the family of glucagon, glucagon-like peptide, GIP, etc. And then you think you can maybe engineer them to become a multi-hormone, become a more potent polyagonist. This was the vision, and have better impact. And glucagon caught our eye, and this was where everybody we knew started to scream at us and say, you guys are nuts, stop it, you're embarrassing yourself. Glucagon, as you know, if you've read the textbooks, is increasing blood glucose. You want to antagonize it, and several pharmaceutical companies are trying that. Why would you want to activate that receptor? It's a bad idea on every level. And we beg to differ. And said, well, if you look at old research papers, there is actually evidence that it triggers lipolysis, it triggers thermogenesis, it triggers energy expenditure. It does a lot of good things, glucagon. It may burn calories. And if at the same time we have GLP, we can engineer a multi-hormone, we can balance the bad stuff it does. So we do want to try that. Well, if you want to try that, you need glucagon molecules to experiment with. And many times it is like it was in this example years ago, that not a lot of glucagon research was done because the molecules available were short-lived, short half-life. They're not very soluble. Nobody really wanted to experiment with them. And what we learned, what Richard learned from GLP and Exendin, I have to press the right button. This one might be it. So this guy here is a lizard called the hairy monster. And in the saliva of that lizard, it was found that there is a version of GLP that has that so-called CEX tail that makes it a more soluble, more long-acting molecule than GLP. And that led to the drugs that now are approved and doing a lot of good things around the world. And we thought, well, maybe we can do the same thing with glucagon. Make a glucagon with a CEX tail, more soluble, longer half-life, start experimenting with it. That was done. And this was the eureka moment of that first initial phase, 2006, where we implanted mini pumps in diet-induced obese insulin-resistant mice and gave them Exendin, the GLP-1 long-acting, and glucagon CX, and had the same effect, decreasing body weight and body fat in ways that looked almost like this could be bariatric surgery. Very different mechanisms, very different receptor populations. So we thought, well, if you put them together, that might really be powerful, and it changed the way a lot of our colleagues thought about this. So Richard made single molecule dual agonists that were, different balances of GLP and glucagon coagonism. This is how this looks. This is published a long time ago. In red, you see 10% glucagon. In blue, you see 100% glucagon. All that is 100% GLP-1, single molecules, once-a-week injections. And these, again, are sort of mice that have been 6 months on a McDonald's diet and are insulin-resistant and weigh 55 grams instead of 25. And they lose a lot of their body weight. It's too fast. You don't want to do this in humans, but it was really impressive. Well, big question now, what do we do to glucose metabolism, right? Glucagon. It's known as the hormone you give in emergencies to somebody who's in hypoglycemia. Well, in this case, in obesity, with the GLP-1 on board, we normalize insulin. We normalize blood glucose. It's not going higher, it's going down. And it really has something to do with glucagon receptors. At least it's not a GLP-1 that's more efficient at its receptor, because if you knock out GLP-1 receptors, you still give these dual agonists, you still have a glucagon activity-dependent decrease in body weight and fat mass. Now, how does that work? Who knows? I mean, you guys working in proteomics are much more aware of the multiplicity of processes involved in biological phenomena. In our field, sometimes pharmacology, clinical medicine, editors of the journals ask you, what's the mechanism? And then they want to have that single pathway. And I don't think that exists usually. But if we wanted to accuse something here, it's FGF21, a protein that increases in circulation. You can treat cells, mice, rats, and humans, as Kirk from a postdoc in the lab did here. And you always, when you give glucagon, increase FGF21. And when you're a mouse and you don't have FGF21, you don't get all the benefits of glucagon on body weight loss and body fat loss. So somehow FGF is part of this. Now, we don't want to treat mice. We want to treat humans. Went into translation. These are monkey studies confirming the result. And last year, the first phase 2 trials came out. Several companies around the world now are promoting these and developing these GLP glucagon dual agonists. And there's better benefits than with just monoagonism of GLP-1. Seems to go in the right direction. Still, lots of question marks. This has to go through phase 3 trials. This has to show that in humans there isn't any side effects, skin, cardiovascular, all the things you may think can happen with glucagon. But so far so good. Now, knowing that almost all obesity drug candidates failed over the last 30 years, we thought it's good to have backups. So we looked at this family again and said, well, how about GIP? That's another incretin, another hormone, comes from the gut, triggers insulin secretion, less harmful potentially than glucagon. So why don't we make a double incretin, a GLP-GIP dual agonist that doesn't have all the potential trouble that glucagon may offer, but gives us more power? Maybe redundant, but GIP goes to some tissues where GLP doesn't go, adipose tissue, for example, and it has very different receptors. So that was done. We call it twincretin. I'm not sure that catches on ever, but we're trying. And that's a dual agonist of GLP-GIP that, to our surprise, we expected great things on glucose metabolism. To our surprise, it decreased body weight, much better than monoagonism. And that went against everything, again, that was out there. Until today, several colleagues around the world think, and it used to be everybody, that you need to antagonize the GIP receptor. A lot of that was based on partially clinical studies, but also the global receptor knockout mouse. And a lesson that we learned over the last 2 decades, a global knockout mouse may teach you that there's something interesting about that pathway and that process, but you can't really deduct you want an agonist or antagonist. For that, you need to do pharmacology. So that seemed to go in a good direction, and this is the paper. Brian was a typical example of that collaboration with Richard, where we had sort of weekly lab meetings and phone calls, and Brian was a PhD student in chemistry, then joined us and became a group leader at our lab in pharmacology, etc. And we found that this works in monkey models and in non-human primates, and it works in humans. Phase 1b trial, in principle, GLP-GIP decreases body weight and improves, of course, glucose metabolism in humans. But the real cool data came out last year. This is the version that Eli Lilly made of a dual GLP-GIP coagonist based on this principle. And they show in a phase 2 trial that was pretty significant, large and long-lasting. Well, there's dose-dependent decreases of body weight and of glucose, HbA1c. That goes way beyond of what a monoagonist can do. This, based on these data, would be best in class and first in class for anything that's out there worldwide. Now, it's difficult to make the right comparison. This is the monoagonist here, but this is a different type of monoagonist to laglutide, a Lilly compound. It's based on an Fc fragment of an antibody. Important, however, is to look at the therapeutic window. In pharmacology, something that you're always aware of. Where are the side effects? Where's the ceiling? How can you maximize benefits? And this is a very telling graph in that series of papers that shows if you compare to placebo and you compare to the monoagonist here, the dual agonist has fewer side effects that seem to be less long-lasting. So there's less of red here, and it's more on the left side of things and compared to the monoagonist. And the point may be you may be able to dose that even higher with better potency to have overall better efficacy and less side effects with the dual agonism, because the receptor occupancy of your targets is less than half, right? And you have the synergy contributing to that. That's the philosophical thing. Now, there are several versions of this out there. And what matters is, do you want to give this once a week or once a day? Is the balance between GIP and GLP best 50-50? Well, the best one right now, the Lilly compound, has a 5-fold more potency on GIP than on GLP. And that's a lot of try and error. And that may seem simple, but if you need to go all the way toward clinical trials with finding out what's the right ratio, that's a money and time consuming process. And you better get it right the second time at least. Right? Now, this is my excuse to be here and my desperate attempt to fit in proteomics. This collaboration with Matthias and his group, where our teams looked at impact of that dual agonist, GLP-GIP, on plasma proteomics, and found a number of interesting things. Impact on inflammation patterns that are always part of metabolic disease, but interestingly, very overlapping looking patterns between what we know bariatric surgery does in humans and what GLP-GIP agonism does in mice, and that would give us full cycle back to our intent to mimic bariatric surgery. Now, there's still lots of questions to be answered, and this is early, but we don't seem to be totally off. Now, if 2 of these are doing great things in 2 different kinds of combinations, how about 3? How about a GLP-GIP glucagon triple single molecule coagonist? Well, that took us 4 years of going back between the chemistry and the pharmacology lab, but we made one. And for those of you who haven't seen them, that's how they look. Very unspectacular. A chain of amino acids where basically you go through every one and say, well, is this one we want from GLP or from GIP, from glucagon? Several of them are shared out, and the ones that are in all together, those are the easy ones. Important, though, that we don't make many changes in terms of introducing synthetic amino residues, because that would increase antigenicity. So then the body thinks there's something foreign, attacks it, destroys it, and you have a number of problems, especially a non-working drug. So only where that's really necessary for fatty acid acylation here, for more albumin binding, or for DPP4, we heard about this today, protease cleavage protection, you want to introduce something that helps, and those are known pharmacological principles. Then you have that CEX tail that we learned from that lizard. And with that, the triagonist, by the way, this has nothing to do with the great hormone oxyntomodulin that was mentioned earlier today. This is the tail that makes oxyntomodulin on top of a glucagon. That's not in there. All right, so if you then look at what that does, this is the famous GLP-GIP coagonist I was just talking about. This is the triagonist. Better body weight loss. These are levels, these are doses where a monoagonist that's currently approved, GLP-1 based drugs, don't do anything. This is much lower dosed. Well, you have food intake decrease, fat mass decrease, the liver is clear of fat, cholesterol is improved, FGF21 comes up when you dial glucagon into the GLP-GIP, and glucose tolerance is improved. So triagonists seem to be even better. They seem to be even better in monkeys as well, and there are now clinical studies that we don't know the results of yet. Now, this is the landscape as it looked a couple of months ago, and that constantly changes. There's always new companies taking up that principle, and others giving up on some compounds that weren't showing the benefits they wanted to see. But interesting, if this continues this way, we hope that in 2 or 3 years, we may have approved drugs that may be better than anything that's out there today. All right, so in the end, we didn't stop there and try to come up with something entirely new, saying, well, you know, we tried hundreds of combinations, and I showed you the ones that worked, of course. Now, we needed a bigger toolkit. So we looked at steroids and small molecules and said, can we put them on peptides? Not only have more possibilities for combinations, they're pretty powerful, but can we deliver them with a peptide vehicle to certain organs and steer them away from others, sort of like a more precision medicine approach, where we can target the liver, can target the hypothalamus, can target the pancreas with certain additional steroid action. And that's how that looks, that you have, we think, that is a movie with our wishful thinking, a peptide receptor at a certain cell, where then this whole thing is internalized, a conjugate of the peptide and the steroid gives off the steroid, it works in the cell. If the cell doesn't have the peptide receptor, that doesn't happen. So you have some cells that won't see action, even though, oh, I have to stop, I'm told. I'm almost done. and you have some cells that see the double action, all right? And we did that with the hypothalamus, we did this with the liver, for fatty liver disease, and with estrogen. And with the estrogen one, we can look at the islet of the pancreas, destroy it with some streptozotocin, and then rescue it with, for example, an estrogen on a GLP. And I stop here with my final slide that says, the vision is an improved metabolic medicine of the future with precision medicine, where some individuals and subpopulations need a double incretin, a glucagon for lipolysis. Maybe they need something that works on hypothalamic inflammation with the dexamethasone, which we have made so far, targeting fatty liver specifically, or need a full triagonist and like full blown out metabolic syndrome. For that, we need, of course, better diagnostics, and proteomics may help us here to know who is who wants to, and who most benefits from these drugs. These are all the individuals that do the work every day while I'm traveling to anniversaries and get a good buffet. We couldn't have done this without the collaborators. I do want to mention Richard DeMarchi, without whom we wouldn't have any molecules. You need a lot of funding for that stuff, and I'm grateful for all of these agencies to fund us. And I hope I did entertain you, even though it wasn't about proteomics. Thank you very much.